we've always thought of cancer as a true genetic disease, and it
Right.
is, right?
Right.
And from Bert Vogelstein and his group identifying APC, as the sort of the initial seminal mutation in many of these cancers, and subsequently getting KRAS and P53 mutations and other things like that. Um, we now know that maybe metabolism is part of this too. So
Hmm.
could cancer be a metabolic disorder as well as a genetic disorder? That's my question. And I think the answer is yes. So we now know that inflammation appears to be present in a tumor microenvironment, and that inflammation likely causes immunosuppression and reduced immunosurveillance. So
Mm.
one possibility would be that when you develop these common mutations in your billions of cells lining the colon wall, and that happens every day, and
Hmm.
they're normally destroyed by
Yeah.
your immune system. They become apoptotic and they go away. But what if you have an immunosuppression locally, a chronic immunosuppression? Would more of those take hold or take foot? And one example of that is a mouse study, and there's been a number of them, but there's a mouse model called the APC min mouse. And in
Hmm.
that mouse model, they basically have a single hit. In APC can have two hits, but one hit in the APC allele, and the mouse is born with that in every cell of its body. And if you give that mouse a highly obesogenic diet, rich in arachidonic acid, rich in omega-6 effectively, they produce many more polyps, 60% more
Hmm.
polyps than a normal mouse. So you're putting that inflammatory milieu in the presence of a mutation
Yeah.
that could cause cancer. And all of a sudden
Yeah.
you're getting more of them, more
Yeah.
hits.
Double
To
whammy, kind
me,
of.
to me, that's kind of de facto evidence that an inflamed environment could lead to increase. I don't know whether it's increased mutability, but increased chance of mutations taking foot.
Yeah. So if I'm tracking here, because I was actually about to ask you, as someone who's never been a cancer biologist, I never studied cancer biology per se, but as someone who's always in biology, I remember as a student sort of just having the notion in my mind, oh, cancer is from mutation. You get X number of mutations in tumor suppressor genes or whatever, and then the normal cell becomes a cancer cell, and off you go. If I'm following you, Tim, what you're basically saying is yes, there are these mutations involved in creating cancer cells, but normally, I mean, there's so many cells in the body getting so many mutations every single day that all of us at some rate is generating, cancer cells. And oftentimes, more often than not, when things are working appropriately, our body takes care of that. Those cells are detected and they are destroyed, and the immune system is involved in that. It sounds like what you're saying, Tim, is that if you're in a certain inflammatory state, if you have chronic inflammation or what have you, then immune surveillance can actually be sort of turned down such that the probability that any one of those cancer cells that would have very likely been destroyed before it became full blown cancer and problematic actually doesn't get destroyed. And so, even though mutations are necessary to generate a cancer cell, the inflammatory state of the tissue can also dictate sort of the probability of whether or not that cancer cell will actually be handled when it should be.
Right. And we call that tumorigenesis, by the way. So, or carcinogenesis or tumorigenesis, basically the increased ability of the metabolic state to enhance tumorigenesis is what we think could be happening.
Mm-hmm.
It's also possible that once you have a tumor, that the inflammation can make things worse.
Mm.
And we don't know whether there's a threshold of inflammation or whether, in other words, you just exceed this threshold of inflammation, it's all bad, or whether
Yeah.
there's a sort of a linear or logarithmic relationship between inflammation and immunosuppression.
Yeah.
We do know that many of the cells in the tumor environment are macrophages, I think about 50%. And many of those are immunosuppressive macrophages.
Hmm.
And we know that the card carrying currency of the TME is lipid mediators. And so having a lot of these inflammatory lipid mediators in the tumor microenvironment likely contributes to this immunosuppression and decreased immunosurveillance.
Hmm. And when you say lipid mediators, is the basic idea here that you've got all of these different immune cells? Some of them can stimulate inflammation, some of them can suppress the immune system. When you say lipid mediators, you mean that these are molecules that immune cells have or they release that are sort of the information signals that are saying turn inflammation up or down and things like that?
I'm going to let Dr. Halad, the expert on, take you maybe through
Yeah.
the pathway from maybe a seed oil that has linoleic acid,
Mm.
to arachidonic acid, to how you get to this
Yeah. Yeah.
these lipid meters and what they actually are.
Yeah.
Sure. Yeah, I like to, kind of glad. Before that, I just want to use the analogy for the chronic inflammation. Like, what happened when you have acute inflammation. It's just like, when there is a fire, firefighters come
Hmm.
very precisely on time, control the fire, and they leave. But in the chronic inflammation, the first thing is these firefighters don't come on time. And
Hmm.
even if they come on, they just hang on where there is a fire and they forget to leave. So the immune cells get in signal, their transit signal and get out signal is totally discoordinated. And that's where the Tim Yeatman's really nicely kind of explain how the immune suppression is developing. So that's the discoordination of that leukocytes that's developing the chronic inflammation. That
Hmm.
is kind of where many metabolic diseases are kind of developing the whole process.
I see.
Now
So,
we'll get
you have signals calling in the firefighters, separate signals telling them when to leave, and if these things aren't properly coordinated, the firefighters go to the wrong place at the wrong time.
a wrong time, exactly. And plus, especially if they are coming late at the tumor site, plus just they are hanging, doing the party with the fats that every day we are taking in the body, and then just there is a continuous progression of the tumor, and that's whole tumor environment we have described in the paper. That's now here is the question of how these molecules are formed. And we know that historically that these fatty acids, they are not synthesized in our body. That's why they called as essential fatty acids. And then those are necessary 1 to 3 to 5%, depending on where we are on the growth curve in our life. Like if we are like early in the adult phase, we need slightly kind of more quantity. And then down the line, as I'm old or, in the kind of late phase life, we may need relatively lower. But what happened to these fats or essential fatty acids? They get used by the immune cells more rapidly in response to acute inflammation and make number of lipid mediators. Same process happen in the chronic inflammation where these fatty acids used by those discoordinated firefighters or the immune cells, where these fatty acids from the diet get converted into the second generation of those molecules. And those second generation molecules we call as lipid mediators or the bioactive lipids. And here we need to note this point that these second generation molecules are more potent than the actual original fatty acids present in the diet. And as you know, and that the standard American diet is enriched with the linoleic acid. That linoleic acid get converted into arachidonic acid, and that further forms the many second generation molecules. And those are the inviting the immune cells. That is the first step of the initiation of the chronic inflammation. So that's how these lipid mediators forms a continuous process that invites more and more immune cells that leads to the tumor progression and kind of continuous growth of the tumor in various regions of body depending on where it is initiating. That is one step. Now, in contrast to that, if you think about, imagine instead of this unhealthy fat, you have the healthy fat, which is coming from omega-3 and other kind of similar related sources, fish or other related sources. And then the same enzymatic system and the immune cell converts those to the healthy molecules. Those are called as a resolution mediators, and that helps to control the immune cells or give the immune cells to get out signals, and there is a clearance of the system and limit the inflammation. So there are totally two arms of the process in the body. Imagine if we have unhealthy fat, then our enzyme system, with the help of immune cells, then that unhealthy continuously kind of feeds the chronic inflammation. In contrast, if we have a healthy fat, then our immune system, with the help of this immune responsive metabolic enzymes, make the molecules, helps to clear the system. So that's how this process, if it is really balanced, then we stay healthy, but if it is imbalanced, then leads to continuous chronic inflammation. But there is a very tight coordination of the fat, what we take every day, and the enzymes present in the immune cells, especially the leukocytes, that determines the biosynthesis of those lipid mediators. And down the line, those lipid mediators, those are very potent than the fatty acids, actual fat in the diet, and gives the clear signal to our immune cells.
Mm-hmm.